So good morning students. Here today we will continue with the next topic, that is the codominance. Next is the codominance. In the last class we had studied about the incomplete dominance, okay, or partial dominance. Today we will go for the codominance. In the incomplete dominance, one gene will be dominant, partially dominant to the other. Okay, but here in the codominance, what happens means, let's see, it is a phenomenon where both alleles, it is a phenomenon where both alleles it is a phenomenon where both allele in a heterozygote expresses in a heterozygote expresses themselves equally. This is the definition of what? Of a codominance. Here you see, it is a phenomenon where both alleles, okay, both the alleles in a heterozygote condition. Okay, for example, this is the capital D and this is the small d. This condition, what we can call? We can call it as a heterozygote condition. Okay, so here in this condition, till now what you studied? This is going to be dominant over this one. Okay, this is the dominant and this is the recessive. Till now we had studied. But here in the codominance, what happens? They both are going to express either that is dominant or recessive. So they are in a heterozygote. They are differing. Different genes are present in an allele. So that we can call it as the heterozygote condition, where both the alleles, this is the dominant and this is the recessive. So this gene and this gene, they both are going to express themselves equally. Okay, they both are going to express and then they are going to produce the new offspring. Okay, so that is the codominance. Let's see here, where both alleles contribute equally to the phenotype. Here you see, where both alleles contribute equally. Both alleles contribute equally means that is in a heterozygote condition, either it is the dominant gene or either it is a recessive gene. They both are going to express their character equally to the phenotype. To the phenotype means to the character. Okay, this is happening in where? During the codominance. So here, to explain this phenomenon, see, once again I will tell you, this is a phenomenon where both the alleles, okay, both the alleles in a heterozygote condition expresses themselves equally, because in a, one more example I will tell you here, that is capital R, small r. This is dominant gene and this is recessive gene. Okay, they both are going to contribute during the next offspring, whatever the offspring they are going to produce. Both the genes are going to express their character. That we can call it as the codominance. So here, one example they are going to take here to explain, to understand this codominance. What is the example? Means that is different types of, see, in the human blood there will be presence of the different types of RBC. Here, example, what they have taken the example means that is the ABO blood group. ABO blood group in human beings. In human beings. This example they had taken. What they had taken? ABO blood group. ABO means there are 4 types of different blood groups. That is A, B, AB and O. These are the 4 different types of blood groups. Okay, which can be seen in the human beings. So here, who is responsible for these different types of blood groups in the human beings means that is the different types of RBC. Okay, different types of RBC. So here, this is an example of what they have taken for codominance. Next is sometimes they will ask for 1 mark. What is the example for codominance? Okay, sometimes they will ask the definition. What do you mean by codominance also? Okay, so here different types of RBC. Different types of RBC determines the ABO blood group in human beings. Okay, here who is responsible for that particular different types of blood group means that is the RBC. RBC which is present in blood, red blood cells. You know what do you mean the red blood cells? These are the main component of the blood, okay, which is present in the blood, and this is only the cells which are responsible for the different types of blood groups in the human beings. How means? Let's see here. This ABO blood group. See, I will explain here. Please note it down the definition and example. Here I will explain. Example, that is the ABO blood group in human beings. This is the example for what? Codominance. Already I had told you. So here, different types of RBC. Types of RBC determines the ABO blood group in human being. Okay, till now, next thing here, this ABO blood group, one gene is responsible for the ABO blood group. That gene will code for that particular trait, then only a human being will have a particular blood type or a blood group. Okay, so here ABO blood group controlled by one gene. ABO blood group is controlled by gene. And what is that gene? That is very important. And that gene is denoted as the I. What is that gene? That is the gene which is controlled for what? For the ABO blood group in the human being. See here, some of the human beings will have the A blood group, some of the human beings will have the B blood group, some of the human beings will have the O blood group, some of them will have the both A and B blood group. Okay, so who is responsible for that different types of blood group means that it is the RBC. That RBC also, how it is going to determine by or how it is going to control the AB and O blood group means that is by one gene, and that gene is known as the gene I. That is by one gene, but here for better understanding we are representing it with I. Okay, so that ABO blood group is controlled by which gene? That is the gene I. See here, on the RBC, this is the RBC. Okay, on the RBC, there will be presence of what? Of plasma membrane. Okay, this is the red blood cell. This is the plasma membrane. On this plasma membrane, there will be presence of some sugar polymers. Okay, sugar polymers means a long chain of sugar are going to protrude from the plasma membrane of the RBC like this. This is the sugar polymer which is extending or protruding from where? From the plasma membrane of what? Of RBC. What it is? This is the sugar polymer. Okay, so this particular sugar polymer, it is controlled by gene. Whatever a chain of sugar molecules are there, that also controlled by gene. That gene is the gene I. Here, this is controlled by gene I. What is this? This is the sugar polymer which is protruding from the plasma membrane of the RBC. And this gene I has 3 alleles. Okay, how many alleles are there? This gene I has 3 alleles. Here, gene I has 3 alleles. How many alleles are there? 3 alleles are there. What are the 3 alleles means? One is IA, another is IB, and the last one is small I. Three alleles are there, and this gene I has 3 alleles. What are the 3 alleles? IA, IB, and small I. Okay. So once again, ABO blood group in human being is an example for codominance. Okay. The next here which determine the RBC, different types of RBC in the human blood determines the ABO blood group in the human being. Next today, here ABO blood group is controlled by one gene, and that gene is represented as the gene I. This gene I has 3 alleles. What are the alleles? IA, IB, and small I. Next, we will see what do you mean by which. What do you mean by IB, IA, IB, and small I? Let's see here. IA and IB, gene allele IA and IB. These are the two alleles. Produces different form of sugar. Okay. For example, this is one RBC, and this is the another RBC. Okay. Here, this is the sugar polymer. This, if they are producing in this way, the sugar polymer, that we can call it, this will have a gene allele, that is IA. And if it is slightly different from the IA, that sugar polymer is slightly different from the IA, that means like this. Okay, it is different from this IA, then we can consider this as the I. Okay, the next see, this is IA and this is IB. One more is there, small I. One more allele is there, that is the small I. So here you see, this is the RBC and this is the plasma. But there is no sugar polymer. There is no production of sugar polymer. Here you can see any polymer? No. If there is no sugar polymer, or the gene small I, this is the gene small I. This gene small I does not produce any sugar molecule or sugar polymer. But this gene IA or allele IA, it produces a sugar polymer which is slightly different. Slightly means here a branched chain of sugar polymer is seen. Okay, so here it is gene responsible by what? By the gene IB. So see here, IA and IB produces slightly different forms of sugar polymer. Okay, they are slightly different from each other. Then small I, the gene small I. This one, this means it does not produce any sugar. So that is this one, small I. This is the gene IA, this is the gene IB. Okay, so the next, because next thing, as you know, the human beings are diploid organisms. Diploid means which has set of chromosomes. You see. Gene alleles. Each person will possess the two genes in which there will be the presence of three alleles. See here, human beings are diploid. Okay, which contains the two sets of chromosomes. So each person are going to possess the two genes. Okay, among the two genes, how many alleles are there? That is three alleles are there. Okay, so here IA and IB, already you had studied, there are three alleles, IA, IB and small. Now let's see which are the dominant one and which are the recessive one. Here IA and IB, these both are completely dominant. They both are completely dominant. See here, are completely dominant over which gene? That it is the small I. And small I is the recessive one. Here, they both are, see, as they have the sugar polymer chain, they both are completely dominant over what? This one, which is recessive one. Okay, which does not have any sugar. They are not going to produce any sugar molecule. So, their IA and IB are completely dominant over which gene? That is the small I. Next thing, IA. Here two conditions are there. What are the two conditions? Means if IA and small I are present. See, IA and small I are present. These are the two genes, IA and small I. In this condition, who is dominant means only IA will express because the IA is the dominant gene. Okay, see here, IA, if they both are present, as I told you, it possesses the two genes. Okay, single gene will not be there because they are diploid. Two genes will be present, IA and I. For example, if you are not understanding, this is the RBC and these are the two genes, IA and I, small I. Okay, in between these two genes, where only IA is going to express itself, but not the small I. Here you see, if they both are present, IA and small I are present, only IA will express. Who will express? IA will express because small I does not produce any sugar. That's why they are not going to express themselves. They are recessive. Okay, because IA is the dominant gene. Okay, here small I does not produce any sugar. The same condition you can see with the gene IB. If IB is present along with the small I, in that condition also, only IB will expresses, but not the small I. See here, one more I will write. One more condition. Here you see, when IB and small I are present, IB and small I are present, only IB will expresses. Because IB is the dominant. Only IB will expresses. This is the first condition, where if both are present means only this one will express, this will not express because they are not going to produce any sugar. Here also, if both are there, IB and small I, only this one will express and this will not express. Okay, then third condition is there. This is the second condition. And the third condition, when IA and IB are present, not small I. When IA and IB are present. What happens at that time? They are present together. Listen here. Present together. This is the condition, this is the RBC where IA and IB, they both are present together. In that condition, what happens? Who is going to express and who is not going to express? In this, they both are dominant, they both will express. Okay, because there is no recessive gene. Here, IA is the dominant and small I is the recessive one. In that condition, only this is going to express. Here also, IB is the dominant and small I is the recessive. Only this is IB to express. If IA and IB, they both present in one cell, means what happens? They both will express. See here, when IA and IB are present together, they both will express. They both will express. Because they both are dominant. Okay. They both will express their own type of sugars. Their own type of sugars. They have the different sugars as I have shown with the red color representation. Okay, that both the sugars are going to produce on the RBC. Here you see, in this condition, what happens means, this is the RBC means, only this kind of sugar polymer is going to protrude. Okay, then no sugar. In that condition, we can say this is responsible here which gene is present, IA. In this condition, this is the RBC and this is the sugar polymer. Okay, here this we can say IB. One more gene is there, but it is not going to produce any sugar polymer. Then we can say only this is going to express, but not the small I. Next condition, here the third condition, where both are there. This is the RBC. Here one is IA and another. Here I will draw. Another is I. They both are present and they both are expressing. They both are expressing in the sense what means they both are going to protrude the sugar polymer, or they are both are going to produce the sugar polymer. Okay, there is no recessive gene. Both the genes are dominant as well as they both are going to produce the sugar polymer. Why is like this? They both are going to express themselves. Till now, in monohybrid cross, we had said that one will be expressing, one will not express. Okay, in test cross, you had studied how to identify. In next, that is in the incomplete dominance. One dominant gene will incompletely dominant over the recessive. Okay, but here codominance means both the genes are going to express themselves. Okay, if both the genes are going to express themselves, then both of them are going to produce the sugar polymer. Why this is going to take place? This is because of codominance. Both the genes are going to express themselves. Why? This is because of the codominance. Okay. Next, RBC, which have both A and B types of sugar. What happens? There are three different alleles. Already I told you IA, IB and small a. Let's see about that one. Here, RBC have both A and B type of sugar. This is the RBC. They have both A type of sugar, that if we represent with the G, that is the IA. One more sugar polymer is there, that is IB. Okay, both the type is there. This is the RBC. If the RBC has both the types of sugar, that is A and B types of sugar, since there are three different types. Okay, since there are three different alleles. What are the three different alleles? If we want two sugars are there, three different alleles are there, how many combinations are going to form means that 6 combinations are going to form. How many combinations? That is 6 combinations are going to form. From where? From the three different alleles and 2 sugar polymers. That is 3 into 2, that is 6. So what are that particular 6 different phenotypes or 6 different genotypes of this ABO blood group? So let's see there. Here, this is allele of parent 1, next allele of parent 2, next offspring's genotype, offspring's genotype, lastly offspring's phenotype. So what will be the allele of parent 1? Here, if it is IA, if parent 1, their RBC will have the IA, then parent 2 also has the IA type of allele. IA, then IA. They both are dominant, IA and IA. They both are dominant. They both will express in the offspring. What will be the genotype? Here it will be IA and IA. This is the codominance. They both are going to express. What will be the phenotype? Phenotype means what will be the blood group. Here in bracket I will write blood group. Here the blood group will be the A. Okay. Then second one, if it is IA, parent 1 IA, parent 2 IB, then offspring's genotype, what it will be? IA and IB. Here also they both are expressing. What will be the blood group means? A, B. Okay, the next one more condition. IA, parent 1, parent 2 their allele or their gene is not going to produce any sugar polymer. Okay, so it will be small I. Here it is I, A and small I. This is the genotype of what? Of offspring. So what will be the phenotype or the blood group? Only this one is going to express, not this one. What will be the blood group? Means the blood group will be the A. Okay, here B is there. That is going to produce the sugar molecule, sugar polymer. That is also down. Both are expressing. That's why the blood group will be the AB. Here it's not going to produce any sugar. It's not going to express. That's why the blood group will be the A. Next one. IB. Okay. IB is there. Parent 2 also IB. What will be the genotypes? IB and IB. What will be the blood group or phenotype? B. Okay, both are expressing. One more condition. IB, parent 1 is IB, parent 2 is IB. Offspring's genotype I, B and I, A. What will be the blood group? AB. A. Next year. One more. IB is there. Small I. IB and small I. There the blood group will be the. Lastly, one more is there. If parent 1 has the gene small I, which does not produces any sugar polymer from the RBC, that means the small I. Parent 2 also same allele, that is the small I. Parent 1 small I, parent 2 also small I. They both will. What will be the genotype here? Small I and small I. They both are not going to express here. So what will be the blood group here? The blood group will be the O. What will be the blood group? The blood group will be the O. Okay. So this is about what? About total 6 different genotypes of the human ABO blood group. But it's not 6, it's 7 different. So this is also one of the good example of multiple alleles. Okay, so that multiple alleles means what? Means more than 2 alleles. Okay, that more than 2 means here in the human ABO blood group it's 3 alleles. Multiple means more than 2 alleles. More than 2 means it may be 3, 4, 5 like that. But here in the human blood group, how many alleles are there? 3 alleles are there. So it is a good example of for what? For the multiple alleles. In the next class, I will explain about the multiple alleles. Okay. Thank you. समझ में